Perfect. So we're live now, and so it would be great if me and Professor Thomas, who is today's guest, you please pin both of us so we have nice podcast to overview at the end of the meeting. Already pinned. Perfect. Okay. Okay. Perfect. So. Hello, everyone. Welcome to the SciCom Synapse. It's a great pleasure to have you all today. So, before we dive in, I want to take a few minutes out, and to tell everyone who joined today about what we do honestly and why we exist. So, you know that feeling when you are sitting in a lecture, staring at a concept feel impossibly complex, and you think, am I even cut out for this? Or maybe you have scrolled through a research paper and felt completely lost through science. Is that your field? Or you are in a big conference and uh, actually want to get some guidance on your career, but you didn't have access, money, or connections. So this is where we've been, and we are here of the students too. That's exactly why SciCom Synapse exists. We are India's first and only completely student-run science communication enterprise registered under MSME, Government of India, built by students, run by students, and most importantly, we work for students like you who joined today. So our name has its all a synapse, a biology. It's a tiny, powerful connection between two neurons. Without it, a signals would go. So we are that connection. Our mission is to bridge the gap between science and society, to take what's happening in the lab, in the journal, inside the research corridors, make it real, relatable, and also accessible to everyone. Our vision is simple, but it's bold. It's to build a multidisciplinary STEM ecosystem where scientific knowledge doesn't just stay in the textbook, it connects people, empowers communities for positive change. So, let me tell you what we actually do. We run different projects, and one of them is the next experiment, which is a podcast project uh, where we take complex subjects and also the journey of a scientist to make it easy and communicable to our students. And then there's Kala Tattva projects where science meets art. It's a science art initiative, and we're gonna launch some flagship mentorship program for especially those who are seeking guidance for PhD opportunities in India and also abroad. So, welcome to the SciCom Synapse. Welcome to our community. So, today our guest is Professor Thomas Pucadyil. It's a pleasure to host today Professor Thomas Pucadyil, a leading cell biologist and currently the director of the Biotechnology Research and Innovation Council, National Center for Cell Science, Pune. He's also a professor at Indian Institute of Science Education and Research, Pune. His research focuses on understanding how cellular membranes are shaped and remodeled, fundamental processes that drive vesicle formation and intracellular transport. His lab is especially known for using innovative minimal reconstitution systems to study these protein machineries in action, combining biochemistry, cell biology to uncover their mechanisms. In today's session, we'll explore his scientific journey, his career, his approach towards doing meaningful research and insight for the next generation scientist. Welcome, Professor Thomas Pucadyil, today.
Thank you.
Thank you so much, sir. So, before we dig in to today's session, my first thing that I would love to ask you would be, and also for the students, that you have gone through being a curious student to leading BRIC National Center for Cell Science. How does that journey felt from inside? Because somewhere we also share our alma maters today.
Well, I mean, it's not a journey that one plans for, obviously. These things happen quite by chance and maybe to a certain extent with a little bit of perseverance. Um, but going from a student to becoming, let's say, to, to a principal investigator, that might have had some amount of a desire to get to being able to run a lab and work on projects that you are interested in. But the rest of how you progress through ladders and reach positions of leadership, I would say I don't think there's any plan to that. It just happens. But at the end of it all, I feel thrilled. I feel very happy. I feel privileged, and it's a great honor to be holding this position to serve the institute and the country.
Mm-hmm. Fantastic. So, if you met your younger self in the college, would you believe that you would become a director of NCCS Pune?
No, absolutely not. In fact, I didn't believe that I would become. I would be where I am, even if you were to ask me this maybe last year.
Mm-hmm.
So, like I said, I don't think many of these things are planned, but I think what's more important is to stay in the game, to be connected, to be
Mm-hmm.
involved. There's a certain degree of, like I said, perseverance, and the rest just happens by, maybe it's like a coin toss. I don't know, but like I said, none of this is planned. And forget about when I was a student. Even till last year, I don't think I would have. If you'd asked me this question, I don't think I would have been able to give you the answer that you find right now.
That's super good. So your work dives into how cell organize themselves. So if you had to explain what you study to a 15 years old kid, how would you do it?
So it's actually a very in many ways it's an area that is intuitively very easy to grasp. It's about how cells contain everything inside of them, even from the most primordial living entity would have something that distinguishes the inside from the outside, and that's in the form of a membrane that encloses everything that is living. And what evolution, if you see through the course of evolution, it's in practically in every life form that we've studied, the one material that cells have used to contain life are membranes, are lipid bilayers. And I can explain more about this. There are a variety of reasons likely why this was a chosen material, but the concept wise, you want a material that is resilient to contain everything inside a cell to sustain life. But then at the same time, you want to form compartments out of that same material, or you want to divide that same material into duplicating life, right? So right there is a contradiction in terms of what you demand or expect out of this material that forms membranes. How is it that on the one hand they're resilient, they're stable to form cells, but on the other hand these cells need to divide, and something has to change about the membrane, or that you want to form compartments inside cells, which is again, if you will think of them as mini cells inside cells. And that's essentially what we're working towards, try and understand how do you form compartments and how do you work against this material called the lipid bilayer or the membrane in order to do so.
I see. I think I remember the cell biology classes that we used to have and the division of labor, compartmentalization, and it was just super beautiful. And the way that you explain, I think That was really fantastic. I hope the audience would really able to grasp it immediately. So, if given one experiment from your lab that would make you go like, "Wow, the biology is wild," what would the experiment that you would choose?
You mean a new experiment or another one of a similar kind that's already been
Or
done?
maybe something that you already did previously, and you felt
Right.
that
So
this is something wild.
yeah. So this essentially starts off with when we had started the lab, we developed assays that would allow us to look at the process of membrane fission, and that's essentially the step which separates two compartments or leads to the
Mm-hmm.
formation of two compartments. If you start with one single contained entity, how do you divide it into two? And that's essentially the basis of how cells divide, how organelles form, and all of that. Now,
Mm,
some of these assays are very straightforward. They are essentially an array of membrane nanotubes that rest on a glass cover slip. And you take this cover slip, you place it under the microscope, and you can visualize these membrane nanotubes. They're literally like, they're like threads in flowing water. Very parallel, long threads, hundreds of microns long, which are all in a
mm,
flow cell. So they are sort of suspended in buffer solution. And if you were to add cell lysates to these, I recall one of the first observations we made was there's dramatic activity that you find in these lysates. They, as soon as these lysates hit these nanotubes, they literally get diced into tiny pieces. And that was our very first observation of fission in real time, and with that degree of
mm,
clarity. Now, the really nice experiment in this context, and I say nice because I've grown up to appreciate and respect the power of biochemistry. And very often, when you start mixing things that are as complicated as lysates with slightly sophisticated assays, then you wonder if is this just some bizarre activity that you find just because you're working with a very complex set of proteins. So
mm,
turns out it's not. So if you start making these nanotubes with lipids that are found in mammalian systems, and if
mhm,
you flow tissue lysase got from a mammalian source, then you see this fission activity.
mm,
But if you
mm,
swap the lipids, if you take bacterial lipids, then you don't see this activity. Or if you replace the lysate with a bacterial lysate and add it to mammalian lipids, then you don't see this activity. So that's
mm,
right there. There's a division of labor in terms of the tree of life. Lysates from mammalian systems act on mammalian membranes, and lysates from the bacterial system act on. So that actually gave us a huge boost in terms of confidence. It told us that there's something that this assay is reporting,
mm,
and that it essentially paved the way for the lab to run screens to identify proteins that could show this activity. So I still recall
mm.
that observation, and it was done with two grad students back in around it was actually 2012 when we first carried out these
Oh,
assays.
I think, this is beautiful, or I would rather say it's a beauty of biology that you can actually observe things happening real time with your eyes, and that just blows out the mind of a scientist. So I could completely relate with you in that sense, and not just me. I think a lot of audience and students as well who are doing experiments by themselves would also definitely agree to this. So you mentioned, your one of the experiment that helped you to kickstart everything. So, what is the scientific question that still keeps you up in the night at this point as well? That this is some mystery that you would love to resolve in your career.
So, like I said, we are a lab that understands or studies membrane remodeling, and collectively, that's a term that's applied to how cells form vesicles. And this
Mm-hmm.
could be vesicles towards the process of trafficking. This is how you move proteins and lipids across different organelles. In terms of complexity, the next level would be, this is exactly the same process by which cells produce organelles, and eventually how cells divide. Now, if you were to understand from what you've learned over decades, is that it's not easy to separate membranes, just because just in terms of pure energetics, like I said, this is a material that's extremely stable, that's evolved to be resilient to these kind of forces. So you've had to likely evolve dedicated protein machineries that can overcome this energy barrier in order to separate membranes. And from, again, what we've known from decades of work, many of these require nucleotide hydrolysis for energy. But it seems we've just scratched the surface, because if you look at the number of proteins that are known to cause membrane fission, you can literally count them on your palm, one palm. It's about 4 proteins. But if you look at the number of compartments or the number of processes that involve fission are concerned, in an average, your routine cell, there are hundreds of them. Now, over the years, we've made knockouts of all of these 4 proteins, and surprisingly, the bulk of trafficking just continues. So there's likely at least many more proteins that manage this, and we have no clues of these. We have no, we don't understand which of these are proteins that are involved in fission. I'll give you an example, and I think maybe the audience will understand this even more so, because this is an old problem. This was the area of secretion. If you recall, we've had lovely screens that are carried out in the East to discover the secretory pathway. This is where proteins get synthesized in the endoplasmic reticulum. They get packaged into vesicles. From there, they move to the Golgi. From there, they release vesicles, which will eventually fuse with the plasma membrane, and that's how you release proteins from every cell. Now, the screens that are carried out picked up a number of proteins that were required for this process. If you affect any one of these, then you essentially don't see secretion. But there is not a single protein that is picked up which, when tested later, causes fission. So we basically don't understand how you form vesicles even in the secretory pathway. And this was, I think these are proteins or screens that are carried out in the 80s. And that's still an open, it's a tremendously complex, and it's a gaping wide open area that would require a great amount of effort and understanding and devising some of these new assays to figure out how this process is managed. So that's essentially where we are headed. We're trying to devise these assays to be able to pick up novel proteins. We've gone up in the list from those 4 proteins that I told you about to 5 now. The hope is to be able to exponentially populate that list of proteins that help us understand how cells produce vesicles.
I see. So basically, there are still missing proteins which are still unknown, and you would love to dig in and find out that what are the exact ones and what are the division of labors and roles of those proteins.
Yeah, absolutely. Yeah.
I think this is a beautiful concept, because like I also heard, I might not be true, but we might have mapped genes genetic, but we still cannot map the complete human proteome. And I think it's been more than 25-30 years that scientists are behind this, but I think we still lack the expression profile of.
So if I may add to that, I think it's a very important point that you raised, and it's I feel it doesn't get said enough. So we have a lot of information. We have decoded the genome, for
Mm-hmm.
both the coding regions and the non-coding regions and the short ORFs and all of that. We
Mm-hmm.
have done a great job with the proteome, and we continue to do so. We go on making that list more and more comprehensive. We've done
Mm-hmm.
fantastic when it comes to mapping cell phenotypes. There are now beautiful
Mm-hmm.
atlases that are available online, open source, that tell you about how organelles are arranged in different cell types, how the cell morphology itself is different depending on the type of tissue you look at. But
Mm-hmm.
what we are lacking in is mechanism and assigning
True.
functions to proteins.
Proteins.
Currently,
Mm-hmm.
the bulk of functions for proteins are annotated based on homology modeling. So
Mm-hmm.
you know a protein that carries out a specific function in organism X. You know the fold of the protein, and you venture to say that every other organism that has that protein carries out that same function. Now, largely,
Mm-hmm.
this mapping, one-on-one mapping, has held true. But there are a number of surprises. You could have evolved functions with exactly the same type of structures and domains. And you wouldn't have been able
Mm-hmm.
to annotate this because no one's gone and then tested these proteins for function. So that's an area. It's called, traditionally, it was always called molecular biology and biochemistry. But I
True.
feel we are moving away from that. We are into big data. We are into AI. But
Mm-hmm.
there's essentially an entire chasm of uh,
Mm-hmm.
of um, of science where we are lacking knowledge.
Definitely. I would say, like, even when we work, even in my system, sometimes we make a knockout and we expect that, okay, we want, when we have these gene knockout, we want cells to behave in this condition. But that turns out that it's completely something different than, what we expected. So I
Yeah.
think, yeah, as you mentioned, evolutionarily, there could be multiple roles of and multiple functions as well, which we couldn't annotate till now.
Yeah. Yeah.
So we discussed the science and also a lot. So a lot of students feel that maybe they are not smart enough for research or these kind of complexities. What actually makes someone good at science or a student good at a science, as per you?
So I think, I mean, I could extend this to practically any profession. I am of the firm
Mm-hmm.
belief that I don't think science is any different from any other profession. Um, I think it requires a certain interest level to begin with, but then that's exactly what you would also require if you were to get into doing business, or if you were to get into doing you know into the fine arts or humanities or any of those other areas. So you need to understand what your calling is. I mean, you don't have to be absolutely sure about where you're going to move towards, but there has to be some sense of where do you think, what excites you. That's important to know. And
Mm-hmm.
after that, it's about trying to consolidate, imbibe, assimilate everything that you learn. And it's science only thrives based on a sense of discovery. So you need
Mm-hmm.
to know how to be able to wade through, you know, tons of information and still
Mm-hmm.
emerge as someone providing or contributing new information. So that will
Mm-hmm.
necessarily require you to understand what is already been done and what is not done. So um, it's some of these small steps which essentially I feel give you the confidence of moving forward in the sciences. It's not about
Mm-hmm.
it's really not about any, you know I can probably say this safely, now that I've spent a couple of years in practicing science. There is a pedantic component, a pedagogical component to science.
Mm-hmm.
which
Mm-hmm.
are what you learn from your classroom teaching. But there is a much larger aspect to science, which is what you learn from working in the lab or in the field. Um, and I don't think I think that second part needs to be emphasized. And maybe if I have the chance, I can explain this. Using some examples when I talk about my research, if there is some time for that.
Mm-hmm. Definitely. So, uh, so you mentioned nicely, is curiosity something you're born with, or it could be trained over the time to stay in science?
So I'm not sure about the training part, but I'm definitely sure that if you provide the right environment, then
Mm-hmm.
you do end up asking these type of questions. Um,
Mm-hmm.
I think the environment does play a big role in shaping how you uh, you know the I think we are always curious. It's just a question of what are you curious about.
Absolutely.
I don't think
Mm-hmm.
an evolved species like us. I have not seen a single person who's not curious. Maybe a little bored, maybe a little tired, maybe a little perhaps there's a bit of fatigue. Uh,
Mm-hmm.
but I don't think you stop being curious. And the real important question is, how do you channelize that curiosity into asking the right questions? So
Mm-hmm.
it's more to do with the environment that you provide with, the challenges that you are providing people with. And I think that curiosity is intrinsic, it's innate. I don't know if you're born with it, but I think you do end up figuring that out in early life. Essentially, that's how you
Mm-hmm.
get to even getting your ABCD done. There is an element of curiosity over there. So there's not a single
Mm-hmm.
person who hasn't learned ABCD or Kha, right? So it's there.
Mm-hmm.
Just needs to be
I
cultivated,
see.
honed, um, but it's there, I feel.
I see. I see. To have a successful trajectory, I would say for a scientist or even as a day-to-day researcher, I think this is one of the most important factor. But what I also feel by working in the lab on day-to-day basis, or many other students like me would also feel, that most I mean, we get success once, but most of the time our experiments fail or something is expected, but we don't see that outcome. Uh, it would, I mean, could you share a failure that changed your perspective towards the science, or rather your approach towards the science that you are pursuing?
I mean, like you said, there's not a failure. If anything, there's more failure. There's more failure around than success. But
Yes.
this one needs to be a little careful of this distinction. It's
Mm-hmm.
and I realize, I think when you're starting out, I mean, I keep telling my students about 90% of what you do on the lab bench, the world is
Mm-hmm.
never going to know about it. The world will not see that. It's only the 10% that will probably go to your thesis. Maybe 1% of it will eventually make it to a publication. But that 1 or 10% wouldn't have happened without that 90%. But more than, I think there's a way of looking at this. I think it's not a failure if you still figure out if an experiment didn't work out the way you wanted it to. If you still figured out why it did not, then
Mm-hmm.
I think that's, and many cases I can understand for very large experiments or very expensive experiments, it's a bit of a problem to figure out what went wrong. But think of an example like this. Since I talked about large projects, I can give you an example of particle physics projects. The CERN project, for instance, of
Mm-hmm.
figuring out undiscovered subatomic particles.
Hmm.
Now, I don't think anyone starts some of these experiments. I don't think there's a failure there, right? I
Hmm.
mean, you run the reactor or the accelerator, and you're bound to get something. But we have come to understand some of these experiments not based on this binary distinction of success or failure. You can redefine some of these experiments by saying you would like to know what happens. Like in a
Hmm.
CERN accelerator, you just bombard heavy atoms or at very high speeds, and you break atomic particles into smaller and smaller entities. And the bulk of it
Hmm.
is, like I said, the bulk, the 90% is particles that have already been figured out. But
Hmm.
occasionally you'll get that 1%, which is a novel particle. But the distinction is you don't design these experiments by saying that I want to find a novel particle. You design it
Hmm.
by saying that I would like to know what happens next out of this experiment.
Hmm.
So as long as you state that clearly, it's really not so much about a failure. It's about experiments that didn't go as planned, or
Hmm.
experiments that produce results that you still don't have an explanation for, which is fine. I think it needs to be cataloged, and many cases that is going to provide avenues for future research.
Hmm. I see. So basically, you completely redefine the concept of failure, but rather to say that these are failed experiments and not a complete failure towards your goal. So, you might have, as you mentioned, a series of failures that would have come across the path of the science. Have you ever felt like quitting science? And at the same time, what was the thought that that keeps you going and also would continue ahead?
So certainly, yes. I think not so much in the last maybe a decade or so, but well, I felt this when I was doing my postdoc, because I was in I mean, I did my PhD in CCMB in Hyderabad, and then I went for a
Mm-hmm.
postdoc at the Scripps Research Institute in La Jolla, California.
Mm-hmm.
And this is meant to be a short stint where, I mean, I wrote after I went for my postdoc based on a grant proposal that I'd written with my postdoc advisor, Sandra
Mm-hmm.
Schmidt at the Scripps. And the objective was to devise some of these assays for fission. And I spent 3 years, and we didn't go very far. And I really didn't have much to show in lab meetings or at posters in scientific conferences. And I recall sitting in my car overlooking so the Scripps is on the coast. It overlooks the Pacific. It's a really beautiful place. And I remember
Mm-hmm.
sitting in the Scripps parking lot overlooking the Pacific and asking myself as to what am I doing here? And it certainly wasn't, I mean, it wasn't that bad. It's not like I wanted to quit or anything, but uh,
Mm-hmm.
I think there comes a time when you want to ask questions seeking purpose. What's the purpose of you spending time and effort? But maybe probably that's the most extreme it's gotten for me in my career. Uh,
Mm-hmm.
but I mean, quitting is something that I think every now and then it does come up.
Mm-hmm.
It couldn't be quitting a job or anything or a responsibility, but just taking a break and doing something else.
Mm-hmm.
Maybe that's also
Mm-hmm.
maybe a milder form of calling quits.
Mm-hmm. Yeah, I see. What keeps you going through everything, irrespective of what you feel?
So, like I said, I think it's that discovery. It's results from experiments that tell you that that they provide a brief. Um it's like a peephole into a very complex design that's at play around you,
I see.
and it makes you makes you humble. It makes you, you know, be grateful for the opportunity.
Mm-hmm.
And I've been working with a team for now 15 years, and it's really a lot of fun. It's always been wonderful to bounce ideas back and forth and see people grow from when they start off in the lab to when they finally
Mm-hmm.
graduate. And
Mm-hmm.
I think if you get to be a part of that journey, I think it's privilege. I think that keeps you going.
Mm-hmm. I see. So you mentioned just recently about, students graduating, students growing. So, what's harder, doing science by yourself or enabling others to do science?
Yeah, so that's a transition one makes. When you're starting out, it's when you're starting a lab, for instance, it's easier to do things yourself
Hmm.
because you never really formally trained in mentoring people. You're always
Hmm.
trained in teaching, maybe to a certain extent, and working
Hmm.
with people. But
Hmm.
mentorship, managing people is a different. It's a different science altogether, and it takes time for you to get there. So when you're starting off, it's really easier to say that, I can just get all of this done myself because I obviously have
Hmm.
been trained. I've done my PhD, I've done my postdoc, and this is the first time in my life that someone has seen it fit to hire me to give me money to do my own research. So why not just take that invitation graciously and then get everything done yourself? So that's
Hmm.
the initial phase when you're setting up the lab. Then comes a middle age when you're trying to understand. You're learning on the job. You're learning how to work with people. You're learning how to motivate them, how to define projects, not micromanage them necessarily, but to show them a path and then let them take that path. Um, then comes the next phase where, uh, and actually, so because you're engaged in involved in multiple things, you probably don't have the time to work on the bench anymore. So then kicks in a more managerial aspect of how to run the lab. So there have been instances where, in fact, there have been instances on which I've acted upon, and you can independently confirm this with students who have graduated from the lab. Uh, it has obviously been. I felt it's easier for me to step in and do the job, and I have done that, but not so much anymore. I think I've also learned, probably become a little wise over the years, and but yeah, it happens early on, and I think it's a good thing, and I think it should be that way. I think people should step in when they're starting out labs, roll up their
Hmm.
sleeves, if you are in a wet lab, and get going, get working. And I think
Hmm.
it's a little odd that the moment you get a job, people just stop working. I don't understand that, and I think
Hmm.
if anything, this is really the time when you should be working, right? Because it's the first time, like I said, in your entire career
Definitely.
where you're working for yourself. So,
Hmm.
and you're not going to have students because you haven't become independent. And why would a self-respecting student join your lab because you're not set up? So
Hmm.
you have you and yourself to rely on. So get working then.
I think this would be a really nice piece of advice for them who are starting, freshly starting, or aiming for something big in their life. So
Yeah.
you mentioned over the time, since now given you are the director, what changes have you seen, in your laboratory, or how do you see the bureaucratic role getting along with the science?
So uh, bureaucracy is there for a reason because it is required to facilitate what you do.
Mm-hmm.
And science, or the endeavor of science, is essentially there because that's our mandate.
Hmm.
And you need the science to lead the way. This entire establishment, institutes, people working in labs, everything's is existing because of science, and that's the primary mandate. Bureaucracy is meant to facilitate doing science, and so exactly why you wouldn't. And as a bureaucrat, it becomes difficult. And don't get me wrong, there have been fantastic bureaucrats who've not been scientists. There are some great managers, and we've learned a lot from them. But it's important to know the scientific process, for in order for you to be able to define these two as separate categories. So I find myself increasingly understanding this role more better. I mean, I still run a lab. There's science going on. I have colleagues here at NCCS as well as ISA Pune with their labs and their scientific endeavors and their questions. But to make all of this work and sustainable, you require bureaucracy. And there are ways of improving bureaucracy. There are ways of making bureaucracy work for the betterment of science,
Hmm.
towards facilitating science. And that's been my endeavor. It actually,
Hmm.
I feel, if you looked at administration and bureaucracy from that perspective, it
Hmm.
really it does make it does make it inviting. There are
Hmm.
scientific methods to a bureaucratic setup, which many people utilize that all the time in labs, in asking a scientific question, being rational about things, being fair, not being blinded, set up avenues where you seek inputs from a diverse set of people before you take the next step. These are all points, if you inculcate them in bureaucracy or in administration, I've seen it works wonders. I've seen it's less tedious, it's less tiring, and it becomes exciting for people doing these jobs as well. So I see myself doing more of that, in trying to bring in these aspects of management into bureaucratic
Hmm.
systems or administration.
Hmm. I see. I see. I think, I feel that, I mean, after hosting a lot of guests and the current generation of the scientists who are in the bureaucratic processes as well, I think the academia in India is really taking great shapes, and I think students would definitely love to continue in academia. And I think it is only possible because of the scientists now also taking care of the bureaucracy at the same time. So I think we are growing as an academician and also scientist. So for students in India, there's often a dilemma, uh, sometimes lacking the stability and also at the same time going ahead for the passion of science. What do you think? How should they be choosing research at that career, at the same time finding the stability?
Well, largely it's the same question that has been asked in any time you take up an endeavor. It's important to know your calling, like I said,
Mm-hmm.
and but more importantly, it's important to be able to define yourself in any profession.
Mm-hmm.
You have to be able to contribute in a manner that is distinct, independent from everyone else. So
Mm-hmm.
the question to really ask is, what is the new insight or the new information or the new approach or new knowledge that you bring to the table?
Mm-hmm.
Now,
Mm-hmm.
if you start asking these questions, and it also helps you choose what lab do you want to do for a PhD. Are you going into a lab which asks these kind of questions? Or are you going into a lab which already has a set of questions defined and you're just following through?
Mm-hmm.
Are you going into a lab which allows you, which helps you learn approaches that could define the next generation of questions? In terms of jobs, are you choosing a job which allows you to define the next set of questions, or are you? So are you at the forefront? Are you at the frontier, or are you just following through? And
Hmm.
they're both okay. I don't think there is any. I don't want to sound like there is a class system between the two. But
Hmm.
if you want to do independent science, then you have to be at the frontier. You have to be asking
Hmm.
questions that have never been asked before. If you want
Hmm.
to still be in academia and contribute, then it is the second category where you can still do a great job of seeing through projects. And like I said,
Hmm.
I think unfortunately the way things are set up, it's a pyramid. It's
Hmm.
there is a there is a pinnacle at the top and everything at the base. And I don't need to mention this to the audience. That pinnacle at the top doesn't it's not floating in air, right? It's just staying there because
Hmm.
of that entire base. So,
Hmm.
but
Hmm.
but ask yourself, what do you want to do? And as a result, you will end up choosing a profession that doesn't there's no sense of missed opportunity. I think, and that's important. You shouldn't be joining an area and feel like, this is not my calling, or I shouldn't have gotten here. Um, so, in terms of, I mean, I might sound a little vague here, but if there's something specific, then I can address this query with a specific example if you have, or if the audience has
Hmm.
questions later.
Hmm. Definitely, sir. So, what is one mistake, or I would say, what are the mistakes that you see the current generation or the young researchers are making again and again? And this is something that could be avoided.
So I wouldn't call it a mistake, but I see it as a trend,
Mm-hmm.
and uh, maybe I'm uh, not old-fashioned, but I've seen these trends come up again and again in the sciences. Um, like I said, there is a lot of information that we have at our disposal, and what we
Mm-hmm.
don't have is knowledge. What we don't have is scholarship. Uh, so you can be a scholar in a specific area, and you can ask deeper questions, and by that you can actually attain success. But I feel right at the bachelor's and the master's levels, or even coursework for PhD, or when you write grant proposals or when you defend grant proposals, the emphasis is on knowing a lot of information. We're not being able to communicate first principles of how the world works or how you set up an experiment. Those are getting sidelined, more so these days because of this barrage of AI and everything that is around us. But native intelligence relies on first principles and understanding how things work, understanding fundamentals. And I think our emphasis is declining towards that area. Our emphasis is increasing towards uh, being more well informed as far as data and approaches is concerned, but we are not we are not we are not getting trained well enough to be able to manage or handle the data.
Hmm. I see. I see. So rather it's the training, and also, like, as you also mentioned before, like the overall environment that you get is, or you rather are surrounded with. So
Yeah.
if a student feels lost in their career path, and as you also mentioned, at some point you also feel that, and I think everyone feels that, what would be piece of advice that you would like to share to them at this point?
Lost, you mean what, professionally?
Like in career, that because sometimes, especially being a PhD student, and also like when I finished my masters, I was clear, but there were many of my friends and also colleagues who were a bit unsure that what would they do ahead immediately after their masters, or let's say now after their PhD, where to go, because the ocean is vast. So what would you like to suggest?
I think if you're feeling that way, you should just reach out to people, talk to your friends, talk to your peers,
Mm.
talk to your batchmates. A lot of this gets fixed at the peer level. It's very important to uh, have that thriving. Uh, it,
Mm.
it it doesn't help to be inward looking, and you can be introspective. That is different from
Mhm.
just not willing to share your experiences with your batchmates. Hello, can you hear me?
Uh, I think we lost you for last 30 seconds.
Right, yeah. So what I'm getting at is reach out to your peers, talk to them about
Mm.
what you're feeling, what are they feeling, inquire about what are they feeling, inquire
Mhm.
about whether they're going through the same phase,
Mhm.
um, inquire about how do they get around it. Essentially,
Mhm.
that, I mean, maybe it's just a fallout of the fact that we're always connected electronically, like I am right now, to all of
Mm.
you. The fallout of doing that is we sort of have maybe lost our native intelligence in talking to people right around you and asking how
Mm.
their day was and asking, what are they going to do next, and essentially just plugged in into the real world. um,
Mm.
I would
Mm.
I would always recommend trying that option out. Maybe you'll get a sense of perspective that way. And
Mm. Mm. I see. So, do you still have mentors in your journey at this point as well?
absolutely, yeah. You always have mentors. In
Mm.
fact, your students are your mentors sometimes. Your colleagues are your mentors sometimes, and
Mhm.
uh, people who you've known for a long time as friends, and
Mhm.
they are your mentors. There's no one mentor. I
Mm.
think there's no one size fits all. You have
Mm.
multiple mentors for multiple, um, I think, multiple aspects of your life. um,
Of life.
and you keep learning from people, and that happens
Mm.
only by reaching out to them more and more.
Hmm. I think many of us students like me would definitely relate to this point that, okay, if we run out to this, then I think this is the right person to ask for. If we run out of something, then okay, I think this is the person who can help us in this part. So definitely agreed. So I think this is one of the last question of the today's session from my side, being a host, and I asked this question to multiple guests who were on the SciCom Synapse we hosted. So it goes like, what is your favorite discovery of science until now?
Favorite discovery, there are many favorites.
Mm-hmm.
More than defining the discovery, I think what I really like about what we do is, um, we call ourselves reconstitution biologists, and by that
Mm-hmm.
we start off with a prescription of uh, how things work. And
Mm.
most cases, that prescription doesn't, it fails. You'll have to
Mm.
chart out a new path, and it helps you understanding areas or aspects about your work which you would have
Mm.
never thought of. But every now and
Mm.
then, that prescription works, and you see you discover something based on first principles, like I said before. And that has been, I think that's quite remarkable. It sort of reaffirms your faith in uh, principles of fundamentals. I mean,
Hmm.
that this keeps happening all the time. By and large, we essentially don't get to where we wanted to, but every now and then we do. So, I mean, making these templates is an example of that kind.
Hmm.
We know how membranes are as far as materials are concerned, and we actually knew how to make these tubes on paper, on these membrane nanotubes, and it was wonderful to see that it came together based on that very preliminary, rudimentary idea.
Hmm. Hmm. I see. So I think I don't know how many students would have joined today from the vesicle biology or reconstitution biology, but definitely, this would be a really growing field because somewhere I also feel that a cell to cell communication also occurs somehow through vesicles, the transport of the cargoes. But if you still don't understand how they are, moving, accepted, and everything, uh, I think it would be also difficult to understand biology that how one cell would communicate in a tissue, and a tissue forms organs, and the full organ is functional at the end, and the end goal is something different, but the cells are doing something on a very very molecular level, and then they communicate across tissues and organs. So absolutely cool. Then I think it was a pleasure. I would like to take some audience question now. So uh, we can choose. Okay, so we have one question from Jyoti, and she says that she really wants to know your journey and the struggles of a student and then now becoming a professor at this point. So maybe she wants to know more about your alma mater, and also how did you start up as a student and uh, then PhD.
So I started off as maybe I should start from my bachelor's. I grew up in Ahmedabad, and I did my bachelor's at St Xavier's. Before that, I did my schooling in St Xavier's Loyola Hall. And my journey into science was not out of my choice. I shouldn't say that. I don't think it was. Um, I certainly didn't know that I wanted to do science, but science was the next
Hmm.
option. Was the obvious option for me, I would say.
Hmm.
So I did my
Hmm.
bachelor's at St Xavier's College, and then I did my master's at MS University, Baroda. Uh, that's Dhairya's alma mater as well. And I joined the biochemistry department pretty much out of um, as an accident, because I had applied for the biotechnology program. This
Hmm.
was the All India Biotechnology entrance test that people would give, because the MS University Biotechnology program was relatively more recent than the biochemistry department,
Hmm.
and it's a centralized exam. So you apply for it, and based on your scores, you get to choose. So I didn't score high enough, and I had got an option in to do. I think it was marine biotechnology in Goa, which
Hmm.
now I feel I should have taken up, but I gave up that opportunity and joined MS University Baroda in biochemistry. But I started liking the course. They have a fantastic. I think the people who taught me are. I think they're remarkable individuals. Uh,
Absolutely.
they they absolutely get you to. I think introspect and understand what your calling is. I think that's their biggest. I would say um, they're experts at doing that, and that's what you should be as a
Hmm.
teacher, especially at the master's level,
Hmm.
because you have to make a lot of career choices at your master's level. So
Hmm.
I finished my master's, and then I finished my first year master's, and then I had the opportunity of doing a summer training, and I went along with a friend of mine. Ashwini, whose house is in Hyderabad. And he said that I'm anyway going to Hyderabad for the summer break, and Hyderabad has CCMB, and I'm going to do my summer training there. So
Hmm.
Ashwini and we just applied to CCMB, and I joined CCMB for my summer training, and I worked with Amit Chattopadhyay, who eventually became my PhD mentor. So, but my
Wow.
summer training work is what got me exposed to actual lab work, and I liked it, and
Hmm.
I wanted to convert that summer training experience into a PhD. So after my masters, I reapplied to CCNB and I got in.
Hmm.
Um,
Hmm.
I finished my PhD. After my PhD, I really wanted to work on um, model membranes, essentially to be able to get into reconstitution biology. I applied to a large number of
Hmm.
postdocs for postdoc positions. I think close to maybe 15 or 17 postdoc
Wow.
applications, didn't receive any response. It is only
Hmm.
Sandy who wrote back immediately saying that, why don't you join us? Sandy's lab is a
Hmm.
largely a cell biology lab. And she was
Hmm.
also interested in trying out some of these biochemical assays for membrane fission. But
Hmm.
this was contingent upon writing a postdoctoral fellowship, which is
Hmm.
what I wrote. And then she said, but she has some money for maybe a year or two
Hmm. Hmm.
while we work towards getting this application sponsored. So that's how I
Hmm.
joined Sandy's lab. And that's where I really started developing some of these assays. In 2 years' time, I also got a fellowship from the Leukemia and Lymphoma Society that helped me go through with my postdoc, or didn't have to rely on Sandy for at least salary support. And
Hmm.
that was 5 years. After 5 years, I mean, I always knew I wanted to come back. I think that's probably because if you spend a significant amount of time in India, and especially at the PhD, at having done your PhD, then most cases it's the I think it's just intuitive for you to consider wanting to come back, which is what it was for me as well. And that was around about the time when ICERs were being set up. This was in 2008 to 2010. And ICER Pune was just being set up, and I applied there, and they gave me a job, and that's how this happened. So um, let me just condense this. I don't think there is, I want to emphasize is, I don't think there ever was a plan. Um,
Hmm.
but at every stage, you do things to the best of your abilities. And I
Hmm.
think, like I said, sometimes things just work out. And like I said, I find myself, or I think I am extremely fortunate for things having worked out the way it did.
I see. Of course, it comes with a lot of hustle, changing countries, continents, and
Yeah.
a lot. But I think
But it's a lot of fun. It's
that
a lot of
looks
fun
like
also, right? I mean, that uncertainty
absolutely.
is, I understand it's unnerving, but it's an opportunity. I mean, how many times are we going to be, allowed this option in life? So it's okay.
Hmm. Absolutely. Absolutely. So I think we would take one last question and then we would love to wrap up the event. And audience can always reach us out for any questions. We would definitely take them to Professor Thomas. Padmaja, is asking, what are the skills you should build as a PhD student or scholar to continue research? Because I think the modern world day is running super fast, the way that it was before, and now it's, I think, completely different. So if you could shed some light on that.
So once again, two things. I don't think you can do research without this. One is you need a firm grasp of basics. And by basics,
Hmm.
I am not talking about biology. I'm talking about physics, math, a little bit of chemistry. Those are very important basics. Um, I don't think I was very good at that, but I realized that I needed to be good at that, and I worked
Mhm.
towards it. So
Okay.
that's important. The second is, I kept saying this, understanding first principles. What I mean by that is your only understanding of how the real world works. So the bulk of it is understood very easily based on physics. A lot of it is intuitive to you based on mathematics. A lot of
Mm,
how materials interact with one another is the insights that you get from chemistry.
mm,
So if you know these, then with the information that is now at your fingertips, you'll be able to take that information and transform it into knowledge. Otherwise, all you're doing is you're just going to get lost in this, and more so now, because at the rate at which information is becoming accessible, we don't need people figuring out that information. We need people being able to help us understand and convert that into knowledge, and that's where your first principles are going to be important.
mm. I see. I would also agree because in today's world, we have a lot of data on the servers available for each and every diseases. I think it would be really rare to not have a transcriptome or a proteome data for even a rare disease at this point. So
Yeah,
I think correctly said that what would we do with that information if it's not getting out as a knowledge.
yeah.
I think this is what it matters.
I think
I
I might
would
also
really
add.
love.
Sorry,
Uh-huh.
I might also add that I think if you have a grasp of that, I think it's going to be very difficult to replace you. You are going to become irreplaceable. Even with an AI, you're not. I mean, an AI is not going to be able to substitute you if you have a
Yeah,
handle of those basics.
absolutely. I think we only made AI, human brain only made AI, artificial intelligence. So definitely we're gonna be ahead of that. I think I would still like to ask you one last question because I love this conversation. The way it was candid and not planned at all, and everything. So most of the students, especially postdocs or PhD students, they are fantastic with the science, but somewhere we struggle to manage people at the same time. So what would be your advice on managing people, especially being let's say a small team when you are doing your postdoc or PhD? Because I think everyone works in a different way, but sometimes it just gets on difficult.
Yeah. So maybe I can just say this in a few points. It's important to know your team. It's important
Mm-hmm.
to know the members of your team. It's important to treat them with respect. They are they are as
Mm-hmm.
much deserving of the respect as a group leader is. And learn to work with people. Don't push people around into getting a job done. It's important to learn to work with
Mm-hmm.
them. Uh,
Mm-hmm.
and I, I think the second aspect of treating people with respect is very important. I don't think
Mm-hmm.
you can run a team if you don't have that going for you. You can always boss around. Um, you'll get work done. You'll get your papers out. You'll get you
Mm-hmm.
know things done in a short-term basis. But one is it's not going to be gratifying. Second is you've got this one life to make a difference, and if you're going to stick it out for a long innings, then it's these are good traits to have, management traits to have. So
Hmm. I see. Because we often see that great scientists or great leaders, they sometimes they also struggle to manage people at the same time. So I think this was about it. So I think we have covered uh, a lot of questions, and I hope, Professor Thomas, you would have enjoyed the conversation uh, today that we had at the SciCom
Absolutely.
Synapse.
Thank you for having me. Absolutely. And like you mentioned, if there's anything that you would like to know about NCCS, ICER, the lab, the work that we do, just write to me. I'll be happy to respond.
That, that would be fantastic, sir. I think I would definitely love to visit NCCS or ISER whenever I am
So,
in India, and would definitely love to be there. Great. So it's a wrap for today's fantastic session. To every student out there here today, thank you so much for showing up, staying curious, coming on a Sunday morning, uh, giving us your time, energy. We genuinely hope that our students and audience are leaving today with some valuable and new ideas and a conversation that sticks in your brain at least for next a few days and helps you motivate, pursue science. So that's all what we want at SciCom Synapse, and make feel science feel a little less intimidating and little more of ours. That's what we do. And also, thank you so much to our fantastic, incredible team at the SciCom Synapse who works to make this happen. Every late night rehearsals, behind the scenes, panic, and what we come up today with the conversation. I think it's the team who takes it all. So, and of course, Professor Pucadyil for accepting our invitation. Fantastic opportunity to host you today.
thank you very much, and good luck to all of you.
Thank you so much, sir. Thank you so much.
See ya.
Bye bye. Have a nice day.
You too. Bye bye.